All right, so let's go ahead and get started here. Welcome everybody. I'll say my name again. My name is Benjamin Allen. I am the outreach coordinator for the KBase project based out of Oak Ridge National Laboratory. KBase, if you didn't know, is the Department of Energy Systems Biology knowledge base. It's a free and publicly available resource for the scientific community to perform all sorts of multi-omics analysis in a nice, simple, and easy to use graphical user interface. And it's so much more. And we're going to talk a little bit about all of the things that KBase is in a second. But the best way to get started is, of course, to go to kbase.us, where you can see our sort of homepage here, where we have different links to information about the system. If you want to find out about webinars or engage with us or learn about the research and the publications that are coming out using KBase, you can, of course, follow these links at the top of the page, and this will take you to where you need to go. One of the key links that I'll point out here is our support page. So we have a lot of different kinds of users, a lot of different kinds of scientists that are trying to do a lot of different kinds of things. And so it's not always, it helps us if you have any kind of question, if you have an issue, if you want to know about a specific tool or have questions about some specific aspect of KBase. We encourage you to go and sign up for our help board and create tickets there. It's a lot faster than emailing me or hitting us up on Twitter, and it helps us triage and diagnose and deal with the different issues that come through from people trying to use the system. So please create a help board account as well as creating a KBase account. It will serve you well in the long run and helps us really improve the system. So if you have not yet done it, go ahead and create a free KBase account. You can do that by clicking the sign up link in the top right corner of the home page. I'll just drop the link into the chat as well here. And the nice thing about KBase is that you can use existing authentication systems. If you have Google or you have OrcID, you can clearly use those to sign in. I am an old school KBase user, so I'm going to sign in via Globus, which is a sort of secure data infrastructure that helps a lot of DOE projects transfer data around. So I'm going to go ahead and sign into my account there. And here we go. This is our first view of the proper KBase interface here. The first thing you're going to see is this thing we call the narratives navigator that has all of my narratives. So we're going to start from what is a narrative, and then we're going to jump back into this portal and talk about some of these other like larger organizational things in KBase. But really everything that happens in KBase happens in the narrative interface, as we call it, or within narratives. So as you can see, I just clicked the new narrative button and KBase is spinning up for me a new narrative here. It usually doesn't take too long. If it's taken a long time to load, you can of course refresh your browser and it will usually knock it over and load up the narrative. So this is a blank, brand new narrative, no data in it, no nothing. So we're going to talk about the different aspects of the narrative and then talk a little about the data and the science that you can do in KBase. But we want to start with the basics here to cover all of the usability and functions and features of the platform, so you can kind of get a sense for how to use it and where you can begin. So first at the top, there's 4 parts of the narrative. At the top, you'll see what we call the toolbar. So this allows you to do some management stuff with your narrative. For instance, I can give it a title here, like Intro to KBase, and I'll hit enter, and there you can see I've renamed my narrative, it saved it, You can see that this one was created by me. If it was created by someone else, it would say their name and their username. And I have some different features that I'll come back to a little bit later. So we have the toolbar. The next section of the narrative is the data section. As you might expect, this is where we add data into the narrative. You can see if I click this big add data button, a slide out appears with some different options for adding data into my narrative. So I see there's a couple of these different sections. I'm going to come back to this in a bit, but just know that this is where you add data into your narratives. This is the only interface that we have right now in the system to do it. So it's very important to know that this is where you get data into your narratives. So I'm closing that. Below the data panel, we have the apps panel. And you can see I've got, with this view, I've got some different categories here that I can unroll and show all of the apps inside of the inside of KBase here in these different categories. And I'll talk a little bit more about apps in a second, but let's just go to the last part of the narrative interface, which is this big section in the middle, which is the narrative itself. And anytime you start up a narrative, it's going to show you this welcome cell that has some helpful links. So let's just show these links just to refer to one of our very useful resources, our documentation page. So if I click any of those links, or if I just go to docs.kbase.us, and there's of course links on our homepage as well, this will just take you to our excellent documentation resource that goes through all of the different basic functions and features that I'm going to talk about today. So if you need to refer to this later, you can access this pretty easily. And yeah, it just has different sections in the documentation that go through all of the basic operating procedures of KBase here. But I'm going to go back into my narrative to talk a little bit more detail about our data and apps. So one of the things that I'm excited to debut to everyone today is that we actually have just released a new feature, and it's so fresh we haven't even announced it to everybody yet because we're doing a little bit of tweaking on it. But we now have the ability to do bulk import. So this is, of course, a very highly requested feature because almost certainly all of you are dealing with large amounts of data. You have lots of genomes, lots of assemblies, these kind of things. So we have just built out a bulk importer here. I was testing it out, so I'm going to actually delete some of these files since I'm going to re-import them here. But within the imports or the data slide out, you'll see I went to this last section here, import, and this is of course where I import data from my computer into KBase. So I'm going to delete these so that I can re-import them here. But this is where you can do, this is what we call the staging area. And so the staging area is sort of like it sounds like, it's an intermediary area between the data that you're pulling into it and the data that goes into your narrative. So when I add this data here, it's not going to immediately put it in my narrative. It's going to add it into my staging area, and then I will add it into my narrative. So here you go. I've got my example data here. I'm advising against dragging and dropping whole folders right now. We're working on an issue with that, but I'm just going to select a bunch of files here, and I'm going to drag them and drop them into my narrative. And you can see it's going to take a second, but all of these files are currently uploading. And once they upload, they're going to populate into this lower section of the import panel here. You'll see they're completing there. And actually, I got to hit the refresh button. And that should do it. Oh, maybe not. I'll go back and then come back in here really quickly. Oh, yes, so I will enable live transcription. Sorry for not doing that earlier. So I am going to, I don't know why it's not. Okay, here we go. It's kicked it over finally. So what you can see now, I've got these, this data that's appearing in my staging area. It's all getting ready to be added into the narrative here. What you'll see is that with some of these file types, it automatically selected what type of file it was. So it says this is a GenBank genome. Others, it needs a little bit of help. So for instance, these are assemblies. So I'm just going to select assembly for these. And you can see it's also checking the box to the left of the file name as I select the type here. Just before I upload, I just want to show a little bit more about what you can see in the staging area here. So this drop down here, if I drop down next to the name of the file, I can see different information about it. I can check the first and last characters of the file, right? You know, if I'm, especially if I, have any issues uploading, it's good to check the headers of, any FASTA or, genome files that you're pulling in just to make sure that they're going to work. But when I'm ready to import all of this, I just sort of select whatever it is I want to import, and I'm going to click import selected here. And what this will do is open up a cell, an importer cell, as we would call it. And you can see it's got two different data types here. In each data type section, I see all of the files that I'm trying to import. And I see some options for naming them. So maybe if I want to remove a file name, oh, interesting. I did not know that. The object name may not be in the form of an integer. So I could just maybe say, or I'll say assembly or something like that. So yeah, you'll notice some of these warnings appear. Sometimes there's lots of illegal characters that you shouldn't be using to name your files in here, and it will certainly give you a big red flag and let you know if you have any illegal characters like spaces or other non-alphanumeric characters. So let's just say I'm happy with the way all of these are named, and I'm ready to import them. So what do I do to import them now? I notice in the top left of the cell, there's a run button. So I'm going to click the run button here. And this will launch a job. So what we're doing here now is something you're going to do a lot of in KBase, which is running apps and waiting for jobs to complete. The whole of the user interface is meant to make running different bioinformatics programs a lot easier. So basically what we've done for any of the apps that we have is create a point and click style service where you select inputs, you set parameters, you name outputs, and you click run. And ideally it just works. Of course, sometimes it doesn't always work, and that's why we have the support board to find out why it's not working or what we can do to triage the issue. But you're going to do a lot of running apps in KBase. That's how we've designed the narrative interface is meant to facilitate much easier use of some of the commonly available public open source tools that are out there. So what you can see now is some of these jobs are finishing up running, and I've got assemblies that are populating into my data panel here. So the files that I'm importing, those jobs are completing, and when they're complete, they will load the data into the data panel here. Just be careful, if you're running a lot of jobs at the same time, if you may run into issues if you start canceling the job, but it's the importer is still pulling it in. Sometimes you'll see this happen where somebody starts an import job, but then realizes they've named a file wrong or something, and they cancel it, and then the job is still running in the background. So just be careful with your batch runs. Do them in sort of manageable chunks, just in case you have one file in 100 that is somehow incorrect. You don't necessarily want to cancel your job just for one file, but if you wanted to, you can, of course. You click this cancel retry all button, and you can select the degree of cancellation that you want to do for your job, or retrying, right? If you want to cancel the ones that are in queue, or you want to stop only the ones that are running, you can select that as well. And of course, you can select which ones you want to retry as well. And so you can see as it's loading these into my narrative, it's changing the count here. I've got 7 successes and 2 jobs running. And so eventually this will say all jobs completed. As I can see, they're all populating in the data panel here. You'll notice with these genomes that I pulled in, it also separated the assembly from the annotated genome object. So if you need to do re-annotation or have multiple annotations, that makes it a little easier. But let's go ahead and talk about adding some other types of data just really quickly. So now the add data button has transformed into a plus here at the bottom right of the data panel. I've still got some reads that are importing here, but I want to just look at some of the other data that KBase has available in the system. So you'll see in the public tab, this takes me to basically all of the public data that we preload into KBase. So you'll see that it's right here. It says the NCBI RefSeq genomes. So these are the full set of RefSeq genomes that are available on NCBI. I can select some other public data as well. For instance, I have the fungal genomes from Mycocosm. That's the Joint Genome Institute's Mycocosm resource. So I can see I've got lots of fungal genomes here. Similarly, there's the phytosome genomes from JGI. These are, of course, the plant genomes that they host. So I can very easily, transfer any of these, big eukaryotic genomes into my narrative just by clicking add and finding what it is I'm looking for. If I needed to check the taxonomy, and then I just click add, and right here at the top, you can see I just loaded this genome into my narrative. So that's one of the pretty cool things about KBase, is that when data is on the system, it's very fast to share it with other accounts, with other users, with your collaborators, whatever. You can move data around really quickly as long as it's in KBase. And we'll show you a little bit more about examples of using this for publishing and using this for sharing your science as well. But yeah, so anyway, this is just to show you that we have a lot of public data that we bring into the system and try to make available for users, especially if you're working on big comparisons or doing comparative genomics work. This sometimes makes things a lot easier for pulling data into the system, so you don't have to go to NCBI and download it all on your computer and do all that kind of stuff. So that's the public data. One thing I'll just show as well, as I mentioned, it's pretty fast to share data or access data that people have shared with me. So that's, of course, what I do under the Shared With Me tab here. And you can see I've got lots of data from lots of different narratives. I can select a specific narrative if I want to, narrow down as I clearly need to here. if I need to select a very specific narrative, I can do that. Or if I want to, I don't know, if I just want to search for something here. Yeah, if you have a lot of data, it ends up being a little slow to build out the list, but once it's there, it's pretty quick. So I'll just do a little search query here, and of course it narrows down the list to things that match this search query. So I can do filtering by types if there's a specific type of data that I'm looking for. So here you can see a lot of the diverse data types that we can host on the system. So this just gives me more options to filter and find what I'm looking for. And of course, I have my data. This is just the data across narratives that I've created, or data that I've imported into the system. So you can see there as well. There's a lot of if you do a lot of work in the system, it can be helpful to use this stuff to filter and find what you're looking for pretty quickly. So okay, that's a lot about data. Let's do something with it. Let's run an app. So we talked about data, and we have the data panel. Let's talk about apps now. So you can see there's a lot of different categories for the kinds of analysis you can do on the system. Everything from basic stuff like you're trying to do blasting, or build multiple sequence alignments, do hammer profiling and searching. You can do genome assembly. There we have lots of popular assemblers like Spades, as well as some unique ones like Hitmer that are based off of some homegrown technology from the Department of Energy. CheckM is, of course, a very popular tool for doing genome quality assessment. So you just see there's a lot of options to do processing on your data. I'm going to show you some tricks that might make it a little easier to find what you're looking for. For instance, let's say I want to just find all of the apps that will take a genome as an input. So in the data panel, I can see my Geobacter urania-rudensens genome here. If I click just below the name, a drop down appears that shows some different options. So I'm going to find this first button here that says show apps with this as input. And if I click that, you can see it adds a little bit of filtering code into the search bar, and it just is going to show me apps that can take a genome as input. So now if I open up any of these lists, you can see, I've got some different options. Let's run CheckM here and see if we can learn more about the quality of this genome. So you can see here it has the input section. I can input either an assembly, a genome, or a set of bend contigs from a metagenome assembly. I'm going to click this, and you'll see that I get a list of all of the data that's in my narrative. So it's just the data that's in this narrative. I click that, and if in this instance, there's not many parameters deliberately, but if there are parameters, you can set them in the drop down. And what we'll usually try to do is expose parameters that are commonly that you might commonly set, and we'll have some things that we've hidden. So, unlike using command line where sometimes you have to, type in every single value for every single parameter for it to successfully run, we kind of pre-program it so that it's, the parameters that tend to not change very much, we hide away so that you'll have greater likelihood of success. So I'm going to click run now. And again, you can see I have just started this app. Right now it says I'm in the queue. And as soon as I'm out of the queue, it will transfer to its running stage. And so I don't actually know how many jobs are running on the system right now, but okay, there you go. It says started running job at this time. So that's good. It means I'm out of queue and now my job is running on our hardware. So if I want to monitor the status of this job, you can see I've got this job status button here. If I click that, I can see that, a little bit more information. I can see how long it was queued for, how long it's been running. And I can even watch the line by line of the job log as it's running. So this is really useful, especially if you just want to understand sometimes how different apps work or like what are they doing along the process. This is a great thing you can watch, kind of watch the grass grow here in the job log, because it will just tell you what stage it's in. If you have a job that's been running for many days and you're just like, what is this thing doing? Is it ever going to finish? You can kind of look into the job log, see where it's at, see if it got stuck at a specific place. And it will, usually highlight significant points in the job log in red, like it is here. It's sort of saying that it has finished different things here, or if it has errors, it will also highlight those lines in red. So this is really useful, especially if you have an error. If you run something and it gives you an error, you can look into the job log, look and see what line did it fail at, and that might tell you, oh, you have this error inside of your file, or this missing, there's often missing information that it will prevent an app from successfully running. So this is just a really helpful way where, we've built this whole nice graphical user interface. This will give you the ability to kind of investigate anything that's happening at the granular level with any of the programs that you're running, and have a little bit more of that kind of traditional control you would have running in the command line, at least for diagnostic purposes. So we're running this app. This will take not too long. As you can see, it was already finishing. So once this finishes, we'll look at the result. But I just want to kind of talk about some of the exciting apps that we have on the system. Like I said, Check-In is an extremely popular one. For genome assembly, right? A lot of you are probably getting into metagenomics research, or you're getting metagenomic samples. You can do metagenome assembly on KBase with MetaSpades or MegaHit, IDBAUD as well. This is really nice because if you 5-10 years ago, if you wanted to do metagenome assembly, you would either, of course, have to send it to groups like JGI or MGREST, or if you really had a lot to do on your own, you might have to get funding to build out a big computing cluster. And part of KBase is making a public resource for this kind of computing power, and giving you access to DOE computing power through a graphical user interface for doing this kind of large scale data processing. So, just as simply as I ran the Check-In map, if I loaded my metagenome reads in here, I can do metagenome assembly, and it will, for like a complex soil metagenome, it will take a couple of days to run, but it will you get access to this compute, and it's a lot faster to do it, and you don't have to sacrifice your own computing power to do these kind of big jobs. In fact, if I can run several jobs, I can close out my browser, I can walk away from my computer, and they're all going to be running there in the background. So we'll often see people do this on the system, where they'll run several large jobs and come back a few days later, and then we see them doing other processing on it. So Some of the other cool apps that we have that are popular, I'll go to the genome annotation tools. Very common to use RAST. This is one of our most popular tools on the system, are these different annotation with RAST. So the Rapid Annotation Subsystems Technology Toolkit. This is just an annotation tool built on the seed ontology that does genome annotation. You can annotate metagenomes, you can annotate isolate genomes, and do re-annotation as well. So we try to keep that database updated so that the annotations stay accurate. But again, it's very quick and easy to do. So for just example, I could either pick a genome to re-annotate or go down to one of my assemblies here. One of these nameless ones here. I can do some taxonomic assignment. In this instance, it's unclassified because I don't remember what it is. I'd have to look back at my downloading history. Or actually, sorry, I should do unknown. Anyway, not unknown marine bivalve. Anyway, you can set the scientific domain. This references NCBI taxonomy as well. So if you know the exact NCBI code for your organism, you can type it in and it will give you that. I'll just say unknown. And I can run this just to give you another example of running an app. So some of the other tools that are new and pretty useful, one is DRAM. This is another annotation tool, and this will do some functional classification as well, and show you, for instance, different sections of the metabolism of the organism. I'll actually show another narrative with that a little later on. Some of the other tools that are pretty useful and kind of unique to KBase. One example here, it's doing this thing where it covers up the bottom. That's annoying. Well, anyway, there is, let me see if I can scale my screen there. We have some viral tools that we've just recently imported into the system. So if you have metagenomes, or you're looking to identify viruses inside of the genome, or look for contamination or things like that, you can use vContact and vir sorter to identify viral sequences inside of your metagenome. So maybe some of you are doing sewage analysis or something like that, and you are looking to monitor the presence of viruses in your sewage. You could use vContact and vir sorter for this purpose. Let's see, what are some other cool apps to highlight? I'll just say, if you're doing any assembly, you'll certainly want to check out the read processing section. Very commonly used tools here are Trim-O-Matic. So if you need to trim out any adapter sequences from your reads or do any reads processing, Trim-O-Matic is one of the easier tools for doing that kind of work. Where you can just set the adapters that you're trying to pull off of your reads, and pretty easily clean them up. So Trim-O-Matic is one tool for that. Another commonly used tool is Bloom filter read error correction. So with Illumina sequencing, this sometimes will help correct duplicates that you see across your reads. And let's see, other things that are useful. If you have reads in the SRA, that's the NCBI sequencing read archive that you want to pull over, you'll see these import SRA file from reads. So you can use this to just drop the URL to the FTP site, whether that's on SRA or it's on another server. You can just drop the URLs into this, and it will KBase will pull those reads into your narrative as a reads data type in the system. Let's see, other things to point out. One of the things that is kind of unique to KBase is, and that we're still developing, is ways to work in batch or to combine multiple data objects into a single object for comparative analysis. So the utilities section is way more important than we really give good time to. There's just lots of instances of tools where you're combining, say, multiple genomes, and you want to build a species tree of multiple genomes. It's much you can mix and match and create new sets of your data using a lot of these utilities tools. For instance, add genomes to a genome set. So what this one will do is I can select multiple genomes that I want to use in some kind of common analysis, and whatever, put them all in here, and then say, okay, I'm going to just say these are Geobacter genomes. What this will do is I'll provide a description. What is it expecting here? Oh, interesting. So here's just a kind of show you all, about some of the things to be aware of when you're running the apps. It will not let me run this until I complete all of the sections. And I realized that I just pulled the wrong tool for this. So this is add genomes to a genome set. So this is expecting that there's an existing genome set for me to add these genomes into, but there is not yet. So what I need to do is go to build genome set. So you would use this tool if you already had a genome set and you wanted to add something to it. But in this instance, I'm just creating a genome set. So anyway, I can select whatever I want to add to it, give it a title here. And so now it will let me run the app. So before here, it is giving me this small red indicator on the right-hand side here that I need to fill something out in this tool before it will let me run it. So if you're trying to click run and it's giving you this crossed out section here and it's grayed out, it means you haven't put in all of the things that you need to do to run the app. So just be aware of that if you're running into that issue. But as you can just see, there's a lot of different things in the utilities section here, merging, slicing, filtering. Just something to be aware of if you're trying to combine or pare down different data. You may find the tool that you're looking for in the utilities section. So just something to be aware of when you're using the system. Okay, so let's just kind of review what we've gone over so far. We've added data into our narratives. We've run apps. We've added public data. Let's take a look and see if our job that we ran earlier is finished. Indeed, it has. So you can see assess genome quality with CheckM. It finished about a couple minutes ago. You can see that the log is complete. I'm going to go now to the results tab here and look at the results. And you can see I've got this nice graphic readout. It shows that this genome is not contaminated. It doesn't have contamination or any strange heterogeneity that's unexpected. One thing to be aware of is this report is showing in the cell here in the app cell, but there's often large reports, especially when you're doing annotation or you're looking at lists of potentially thousands of hits or something. It can be challenging to kind of view this in the app cell. So with a lot of these tools, you'll see view report in a separate window. And like it sounds like, what this will do is open up a separate HTML instance of the result, so I can have a little bit more flexibility with looking at the information. So in this instance, I can look at the table generated by checkm that tells me the completeness and contamination of this example genome. So that's what a data output report looks like. If there are, for instance, annotation tools or assemblers, they will create data objects. So if there was data created, it would show that in the object section here. And it says no objects created there, as you can see. And there may also be files that you can download for use outside of KBase. So something to always take a look at is the files section of your app after it's done. So for instance, this has the plots generated by checkm and all of the output files. So this is an important thing to kind of understand with KBase, is that not all file types become data types in KBase. With anything, any data that we pull into the system, we convert to data types and data type specifications that are specific to KBase. So for instance, you can see that this genome is, the KBase genome type 11. And what this means is that effectively, it means we have interoperability of our apps against different types of data, different versions or different types of the same kind of data. So for instance, there's you may have a GenBank genome or a GFF plus FASTA type genome. When you pull those into KBase, they get converted to a genome object, and that genome object is compatible with these different apps. That we build the apps so that they'll have compatibility against heterogeneous file types, but they may be heterogeneous file types, but they're a singular data type. So that just helps us facilitate interoperability of these tools across the system. So all that to say, some apps are going to generate data, and they're going to create new outputs that are match KBase data objects, and they're going to populate in your data panel here. And then some files some apps are going to generate files that you can then download and use off of the system. So that's sort of the intention of this, is that there's often reports or images or things like that, lists, CSV files that you can download from KBase and use externally. So there's just to say, there's some data that doesn't always appear in this data panel. So that should cover the output of the app there. I know we're coming up at the top of the hour, and like I said, there's a lot to cover with KBase sometimes. So you may find it helpful to reference the documentation after this, but just two more things that I'm going to show in the narrative before I jump out into some of the other parts of the system. One thing you'll notice I'm doing here is, so as I've been building out my narrative, you can see dropping lots of apps in here. It can get a little messy. So I can collapse cells here. So I'm just going to collapse all these cells, and then everything looks nice and orderly. If I need to move things around in the order of the list, I can move cells up and down. If I want to delete a cell, I can also click this 3 buttons here, and this will bring out some options to delete the cell. One thing you'll notice here that's interesting is this show code. feature here, and that actually won't, I don't have anything in it, so it won't show any code. But if I click the check-in tool that we just run, so this is showing the run code for this specific app. One thing you may notice in the the bottom right-hand corner here is this first button here, and I'm going to click that. And what that does is it opens up a code cell that I could, for instance, if I wanted to run apps outside of the graphic interface, app cell style of doing it, I could, copy this code and execute it, and it would run this app inside of it is now, it doesn't show it, but it's running this app inside of the narrative just based off of this code. So what does this mean? It means that you can, if you're an advanced user, create scripts or loops, or you can configure these tools to access some of the program libraries inside of KBase using code cells. And you can see here, this is of course in Python. The narrative interface, all of the ability to kind of have this combined app and code cell in a singular interface is because KBase is built on top of Jupyter, the Jupyter notebooks as they're called. They used to be called IPython. So if you're familiar with Python, you can do scripting and coding inside of this interface alongside using the pre-built apps. One other thing to also mention, next to that code cell button, there is the Markdown cell button. And this just allows me to do commentary inside of my narrative here. I can just write whatever I want, and it will appear inside my narrative. So I'm going to show some examples here shortly of published narratives that use these commentary cells, these markdown cells, in combination with the apps to sort of create basically a living publication of sorts. Like, right, the value of KBase is that as you're using all of these apps, you're building out your analysis, you're basically creating a digital lab notebook that allows you to serve up all of this different information. And so I'm going to do a plug for folks to come to our the webinar we're going to host in 2 weeks, where Zach Crockett on our staff is going to demonstrate using these features for building for doing publishing with your narratives. So how do you use the narrative interface and the narrative publishing features to share your work in KBase with your colleagues, with the world? So I'll just show a little bit of that here. At the top in the toolbar, what you can see is this share button. And if I click the share button, I see some options for managing sharing of my narrative. So let's say I want to share this narrative with a colleague. And so this will mean that KBase user can come and access the narrative with a certain level of permission here. So I'm going to share it with, Zach here. So I found his name. I can see there's different options for sharing this narrative and giving permission. So it can do view only, meaning that he can see everything I've done, but he can't touch anything. Or you can have edit and save, meaning he can make his own edits, add his own data, run his own apps. Or I can even give him edit, save, and share, meaning that he can also freely share this with whoever. I'm just going to do view only there. And now I can see Zach has access to this narrative. If I want to remove him, I can, of course, click the remove button. But there is also this feature at the top. Let's say I don't want to share it with specific people. I just want to share it with everybody. I can make a narrative public here by just clicking the Make Public button. I see my narrative is now public. meaning that I can send this URL of this narrative to anybody, and you can come and look at it. So there's nothing particularly substantial in this narrative. I'm going to jump back in a second and just show some of the tutorials and other resources that we've got that are kind of, premier public narratives. Just want to check the chat really quickly. Okay. Thanks, Zach and Ellen, for responding to folks. So this is what I've just introduced you to here is the narrative, and how do you build out analysis in KBase? How do you import your data? How do you run apps on your data? How do you share your analysis with other people? I'm going to jump back into the previous section here. So narratives, the narratives navigator here. So I just showed you one narrative, but if you're using KBase a lot, you're going to create lots of narratives. As you can see here, I've got a ton of them. And this interface, the narratives manager interface, the narratives navigator, as we call it, this helps you do some management of your narratives, to organize them, to basically try to keep it all together with your work on the system. So when you're in this view, if you want to do sharing, if you want to rename, if you want to, I haven't talked about organizations yet, but if you want to link a narrative to an organization, you can do all of that in this management interface here that we call the narratives navigator. Again, I can also see those narratives that are shared with me. So, for instance, this is this is a project that I'm working on with somebody. So I can open up this narrative, and we're basically working back and forth together on it. And you can see where I've built out this analysis here using different tools. And so it's just very easy to share these things back and forth, to work with, if you have students you're working with, or you're working with a classroom, you can of course use these features to kind of manage access and, make sure that the researchers in your lab are doing things at the same way, using the same protocol, etc. So I just would encourage folks to think about the power of collaboration here. With Google Docs, all of these kind of cloud services that we're used to now, this is very common practice. We're trying to kind of introduce this in the omics and bioinformatics space, so that there is a common platform that people can work together on across collaborations. I didn't do much, showing you all any detailed workflows or anything like that today. We're going to have lots more webinars this year where we do that. And like I said, there's a lot on our YouTube page, where we've shown some different workflows. But when you're in the system, and if you want to look at some tutorials and look at some examples of using the system, just go to the tutorials section here in the narratives navigator, and this will show you all of the different tutorials we have. For instance, drafting isolates or extracting genomes from metagenome annotated genomes from metagenome sequencing data, building metabolic models. There's a lot of different workflows that would take many hours to kind of talk about on their own, that you can go and explore on the website in the tutorials section. Like I said, we're going to do some of these webinars later on in the year where we show greater detail about doing some of these workflows. But if you want to look at it now, this is a great resource. And you can also see what narratives people have made public. So I'm just going to do a example here. So for instance, this is where a user is sharing data from the web of microbes. So these users are all putting these public narratives here because they want other people to come and look at what they've got, whether it's a genome announcement, or just a large public data set. People are trying to share these narratives and make them public to give you and the other KBase users access to that. So you can see there's 1,200 shared narratives here. So there's quite a lot. You can do some filtering, of course, as well to basically find the narrative that you're looking for, and share it with other people. So for instance, here's where JGI has been serving up some of its metagenome annotated genomes in these different categories here in these different environments. So you can access these, download the data to your own narratives, do your own analysis on it. So this is just one of the ways that people are sharing, using KBase to share large data sets with user communities. Real quickly going to go through some of this other stuff. I know that there's probably some questions in the Q&A that I'll get to in a second, but just want to make sure we cover all the basics here in the time we've got left. I know I'm close to the top of the hour here, but so you may have seen some reference to organizations in the interface. So organizations allow you to take a set of users and pull them into a common org where they can then have access to a bunch of narratives that have been shared with the org. So just for instance, this is a project that I've been working on with some people, and so here we've shared the list of all of the narratives that we've been working on together. Anyone that's in this organization can access the associated narratives. You can do some degree of permissions inside the organization as well, so that you can show that they're there, but people still have to request full access. So there's different levels and permissions that you can give inside of these groups, but if you have a project or a whole lab working together, this is a very easy way to bring all of your work together into a common environment and keep it all organized. So you can see I've got the list of people here that are users. I can invite other users, and I can see all of the work that we've done and access it from there. Catalog, this just shows a full list of our apps. So if you want to see exactly, and we have quite a few apps on the system these days, I think something like 300. You can see a lot of information about each of these apps. For instance, you can see how many times it's been run in a narrative. If I click this button here, it will actually open up a app page or a documentation page going over all of the functions of the app. What does it do? What does it take as inputs? What are the parameters? What do they mean? All of that kind of stuff is in the app page. And I can see other useful information too, like the success rate or how long did it take to run. So all of this you can access inside of the catalog here. If you're running a lot of jobs, the jobs manager is a really useful feature, a really wonderful feature that we have that helps you monitor everything that you've got going on in the system. So especially when you have multiple narratives and multiple jobs running, this helps you look at all of that outside of look at the status of these things outside of the narrative interface. Longtime KBase users will know if you have tons of narratives open on your browser, it can be a little slow. It takes a while to load all of that information through the browser. So this is a much easier way to monitor your jobs instead of having to open up every single narrative and look at every single individual job. I can see these are obviously the jobs that I just ran in that example narrative. If I click the I here, it will bring up that job log just the same as I would see it in the narrative. And again, if you have a lot of things generating errors, you can, of course, reference this in any help tickets that you make, which makes our life a lot easier for diagnosing problems. So yes, definitely check out the Jobs Manager and Jobs Browser, and I think you'll find it a really helpful feature. Finally, just want to show the account section here. This is where you can, of course, update any of your information, or if you have multiple sign-in accounts, you can link them together here. We often have people will forget an old account. They made an account a long time ago and they can no longer remember what their login was, but they have data in it, and so you can help link old accounts to your new accounts using this feature as well. So we're at the top of the hour here, and I've gone over quite a bit. I know this is a lot of information. I just want to check and see if there's any questions in the Q&A document that I can answer here that may not be answered. So it looks like folks have been answering those questions here. Let's see here. Yes, we will. Yeah, definitely check out this metabolic modeling webinar if you want a demonstration. We're gonna probably in March or April, we're gonna do some of these advanced workflow tutorials again, like metabolic modeling or metagenomics analysis. Can the codes be edited on the interface? I'm not sure what that one is asking. Okay, can the code be edited in the interface, perhaps? So if you're asking about editing app codes or like building apps and altering any of the programs, so we're an open source platform, meaning that all of the apps that are in the system are hosted on GitHub. And so if I go back to the catalog here, I can open up a example. So you can see in an app, where the app is hosted. Like you can go and look into the code for any app here. So this is the master branch for this FastQC tool. So you can see all of the code that goes into any app. You can, and we'll have an SDK training later on this year as well. There's a video on this on YouTube. That's software development kit. We do have a software development kit that allows third-party developers to build their own apps, bring them into the system, and test them, and so forth and so on. I'll actually show you. So you could not say, you could edit some of the parameters. You couldn't really edit the actual code inside the narrative interface. That is, you can just change parameters and things like that. But if you are an app developer, in our documentation page, you can find information about software development. But if you are a developer and you want to use KBase as like a building and testing environment, you can do that. You'll see in the apps panel here, I can toggle between release and beta versions of apps. So we have multiple versions of apps. We have apps that are in beta that are not, you have to kind of acknowledge that they're maybe not in full release mode. So they're not exactly guaranteed to work because, people are testing them out, but you can try out beta apps on the system if you want to this is a good way to also see what new apps are coming out soon, is to go to the beta apps and see what's in there. And this will show, there may be the release version of an app and then the beta version of an app that is, going to come out soon, but we haven't fully released it. So you'll see a lot of duplication of the apps that we've got in the released app section. But so anyway, that's just to say, you can't change the code of an app inside of the interface or anything like that, but you can build your own tools and deploy them in the system. So that's hopefully that answers that question. Yes, can I tell you the specs of the supercomputer? So it's changing all the time, but what I can say is that we have two main hardware nodes. One is based at Argonne National Laboratory, and one is based at Berkeley Laboratory in the Bay Area. It's several thousand cores are available, across something like several hundred nodes. So we're always trying to add more compute power. We're always trying to upgrade the system to be more robust and to be able to handle an increasing number of users. But it's Cori and Magellan are the names of the two supercomputing clusters that they're based in. We don't have all of those clusters assigned to KBase, but so those are based at the Cori and Berkeley Lab and Magellan at Argonne. So you can look up more about those supercomputers on the lab websites. So I was seeing a lot of vContact and VirSorter questions. There is also a previously recorded webinar on the YouTube page. Let me see if I can find it here quickly. Viral tools for KBase. I'm just going to post the link to this into the document just to make it easier to find. But so this covers using all of the VirSorter, vContact, and other viral tools, and that's with Ben Boldock, who is one of the developers of these tools. So if you are a developer and you build a tool in KBase, we're of course happy to let you take over our webinars and show all of our users how to use the tools. But would definitely encourage anyone interested in these viral tools to check out this YouTube tutorial. All right. So it looks like we're about 10 past the hour. I think I'm going to wrap it here. The last part I'm going to show just before we go, and would encourage everyone to go ahead and sign up for our next webinar on publishing narratives. I only briefly touched on this today because I know that Zach is going to go into a lot of detail about it in 2 weeks, but would really encourage folks to sign up for this one. I'll just go ahead and drop the link into the chat here. I'll just show a small example of this really quickly. So you can, of course, see all of the publications that come out using KBase on our research page. This is just an example at the top here of, that link is broken, clearly. So just wanted to show this here. So this is a static narrative. So we talked about public narratives, but we also have static narratives on the system. Zach is going to teach us how to make these and what they're all about. But just really quickly, what this person did is that they have a publication and they've linked to their narrative in their publication. It's just something we really encourage you to do if you publish with KBase, is to make your narratives associated with your work public and to link them in your publication. But this person has created what's called a static narrative, which is a link to an instance of their narrative where anybody, regardless if they have a KBase account, can come and access this content. You don't have to have a KBase account. You can just link this URL, and that will serve up a rendering of the narrative that gives people access to the data. You can see any tools. This one is just serving up data, but static narratives basically allow you to take a snapshot of your narrative with all of the data and apps and commentary, etc., in a specific state, and then share that current status, that snapshot, with anybody. So what's interesting about that is that you can, of course, you can have a version of your narrative where you've done your work and it's ready for publication. So you create the static narrative to link to your publication and to share with other people. But right, you may want to keep analyzing that data in your narrative in the background without having to move it to another one. So this allows you to keep making changes, making edits, doing continued analysis with your narrative, but you can share a version of that is static. It's a snapshot that is in the state that you want to share it with other people. So that's just kind of a cool way to make a fork of your work and share it with people. But to find out more about that, definitely come to our February 16th webinar in 2 weeks, the same time in 2 weeks, where Zach is going to show us lots of examples of people who have published their research with KBase and teach you more about best practices for building out your narratives and sharing them with everybody. Okay, I think that is that. So thanks again, everybody, for attending this webinar today. I hope it was informative. I hope you learned a little bit about the basics of the system. I know we didn't get into too much details about doing specific types of genomics or metabolic modeling or stuff like that. I was sure you were going to have more trainings throughout the rest of the year, but hopefully you're feeling a little bit more informed about the interface, the different features, and at least you have some good resources to get you started using the system. So thanks everybody for attending, and we'll see you hopefully in 2 weeks.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.